Respiratory therapy students and medical students are commonly taught as an integral part of their education the physiologic concepts governing the binding and unbinding of oxygen to oxyhemoglobin. In addition, we've all been taught about the shifts in the position of the oxyhemoglobin dissociation curve that are triggered by changes in hydrogen ion concentration and or arterial carbon dioxide tension and or patient temperature. These concepts are usually taught in qualitative fashion. But in this 48-minute module, and only here, you will see these changes portrayed quantitatively, which will allow us to clearly evaluate the effects of curve shifts on oxygen delivery to tissues. The presentation begins with a description of inverse and direct relationships, with particular emphasis of the nonlinear direct relationship between oxygen saturation and oxygen tension. Next, the direction of the shift in the oxyhemoglobin dissociation curve, or OHDC, that spontaneously occurs secondary to changes in hydrogen ion concentration, carbon dioxide tension, or patient temperature, either singly or in combination, are described. A mathematical model of gas exchange is developed and applied next to calculate the percent saturation of hemoglobin precisely and accurately. In a spectacular video that is accessible from the website of the American Journal of Respiratory and Critical Care Medicine, we will watch the distal tip of a Swan-Ganz catheter as it traverses the vena cava, the right atrium, the right ventricle, and the pulmonary artery, clearly visualizing these anatomic structures as the beating heart propels the catheter lungward. Real-time video portrayals of human physiology and the precise mathematical models of gas exchange can convert concepts that might otherwise be formidably complex into visual images that convey the intricate inner workings and intrinsic beauty of the marvelous machine in which we all dwell. The intricate structures of the respiratory and circulatory systems are explored in this presentation on both a macroscopic and a microscopic level. Striking photographs and electron micrographs depict normal anatomy as well as the deranged anatomy that results from cigarette smoking. We'll encounter the pulmonary alveolar macrophage, the garbage collector of the lung, up close and personal, and watch individual red blood cells squeezing through pulmonary capillaries, single file. The module then focuses on the characteristic sigmoidal shape of the OHDC and describes the amazing insights of hemoglobin physiology described by G. Richard Kellman more than a half-century ago. The module concludes with an examination of the effect of shifting of the OHDC at the cellular level, both in an acidotic adult and in a fetus in utero. At the conclusion of the lesson, we will be provided with compelling evidence that, contrary to what many of us have been taught in respiratory therapy school, adult hemoglobin is actually a far more efficient oxygen delivery vehicle than its fetal counterpart.